Hello, Anthony. I think you're muted.
Hi,
Yeah.
Miguel. How are you?
Good. How's it going?
Good, thanks. Yeah, holding up despite some Wi-Fi challenges this morning. Wind's blowing, and my office Wi-Fi went down about 5 minutes ago, so I've had to
Oh,
hastily
that.
move venues.
Yeah, it always has to happen like right before
Of
anything
course.
important happens.
Yeah. Hi, Andreas.
Um,
Um, uh, Miguel, can I share my screen?
yeah, I was just gonna ask you. Thank you.
Thank you.
Yeah, let's check that everything works fine while we wait for more people to join. Okay, nice. Yeah,
Can you see my full screen?
yeah, perfect.
Super.
So are you based in the UK?
Based in the UK, you're down in Sussex, are you? In Brighton, near Brighton, are you?
Yeah, yeah, right.
I'm not
Right
too
now I'm
far
in
away from you.
Barcelona, but
Ah, okay.
for a
I,
week, just for a week.
oh, wonderful. Excellent.
Yeah.
I'm quite close to you in Brighton. I live in the Surrey Hills on the Surrey-Sussex border, about
Is that
30
like to
miles.
the is that to the west or east?
It's kind of from Brighton. Yeah, it's slightly west northwest. It's primarily north.
Okay.
Uh, so between Guildford and Dorking, if that makes any sense. Or
So bad with these like small geography towns.
yeah, well, it's
No, ha Hassocks, Burgess Hill, where the train goes mainly.
oh well, you're missing out if you've never been to Leith Hill, which is where I live. It's supposedly the highest hill in the south of England.
Oh, really? Okay,
Yeah,
nice.
in the forest, believe it or not.
Oh, okay. Oh,
Yeah,
interesting. Nice.
yeah, it's about an hour outside London on the train.
Okay, good. Let's see. Okay, I see that Carl has joined already. Um, So yeah, I guess then we can start. So Anthony, feel free to start whenever you want.
Thanks, Miguel. Delighted to be here. Perhaps just briefly about me. I'm an engineer by background. I spent about 10 years in aircraft engineering, avionics, radio and radar systems, studied cybernetics control engineering, and then had a rather circuitous journey as an entrepreneur, building companies in the tech space and then in the tech biotech space. For the last 10 years, I've been looking at trying to model microbe, and microbiome host interactions using software network science. And actually, I stepped back from that about a year ago or year and a half ago. And for some strange reason, I spent the last year just trying to forge those two kind of components of my background. And that took me towards trying to understand more about the mitochondrion. Um, so I'll take you through a very brief introduction to the mitochondrion for those that are not familiar, and I apologize because this audience is probably very familiar with mitochondrion. But mitochondria, in contrast to the way they're often portrayed as kind of baked beans, they're highly dynamic organelles within the cell, merging to form strings through a process called fusion and splitting to maintain cell health, a process called fission. They're perhaps between hundreds and thousands, possibly tens of thousands, and by some estimates, very much more than that, mitochondria within each individual eukaryotic cell. So just a very simplified, stylized model of the kind of organelle under observation today. Here is a notional stylized mitochondrion. In cross section, you see these so-called invaginations, and they're about 40 nanometers spacing between those invaginations. And they're called cristae, individual crista, plural cristae. And it is within the cristae that a lot of exciting stuff happens within the mitochondrion. The cristae are attached to the inner membrane of the mitochondrion through a connecting site, which I'll describe in more detail as we go through the presentation and the conversation. And that also provides access to the outer membrane. There's a lot of activity at that interconnection site. So if we take a look at the crista, that's the focus of our conversation, our attention through the course of today's presentation. The particular cristae that we're talking about are mammalian cristae, and they have at least one type of cristae called lamellar cristae. These are, in fact, less like invaginations and more like flat sacs. And that's interesting because they are extremely regular. Here we are looking at an image of the string of mitochondria. Those perpendicular striations are, in fact, the flat sac lamellar cristae. And it's within these cristae that within these microdomains that ATP synthesis is performed. So we were looking at a healthy mitochondrion there. We're looking now at a dysfunctional mitochondrion, where we see that the regular cristae structure has been lost, and this so-called onionation is a pathology, and it's to be avoided, and structural integrity is compromised, and these mitochondria are soon to be euthanized through apoptosis. So just by way of introduction, explorations to characterize the function of the mitochondrion within the cell have largely focused on what everyone knows from O-level biology, that they are the energy current, they generate the energy currency of life, ATP. This, as many people are saying now, radically understates the role of the mitochondrion in cell organization. It's conjectured here, and this is a kind of a theoretical model of stacked conjectures I'm going to be presenting, that it provides clock coordination and computational functions using what we would consider well-characterized physical principles. The novel framework here posits that the nanomachines on the inner mitochondrial membrane function as part of a complex phased locked loop architecture. Mitochondrial cristae act as resonant cavities, electron transport chains function as phase locked loop voltage reference oscillators rather. ATP synthases serve as driven voltage controlled oscillators and demodulators. And through an array of coupled forces, these components of the mitochondrial phased locked loop architecture are highly regulated and coupled with the Krebs cycle, which runs in the very core of the mitochondrion, the matrix. So why is this necessary? Why is it important to look at this? The mitochondrion is an extraordinarily well-characterized organelle. However, it's fair to say that that characterization is primarily biochemical and electrostatic in nature. The extraordinarily well-characterized function is description of sequential enzymatic reactions leading to ATP synthesis. It's foundational. It was birthed by Peter Mitchell in the 60s with his extraordinary breakthrough, the discovery of chemiosmosis on the mitochondrial membrane. But it does not adequately explain the efficiency, dynamic adaptability of mitochondrial output in conditions of high flux, or the coordinated responses observed in complex cellular signaling. And particularly, it doesn't explain the elaborate, highly conserved, symmetrical and mirrored architecture of the cristae, which appears, to my mind, to be functionally integral beyond merely increasing surface area. Nor indeed does it explain the rapid non-diffusional signaling pathways implicated in processes like apoptosis, calcium handling, and redox signaling. So then on to the novel framework. As I've said, we propose here that the mitochondrial cristae act as complex self-organizing within a complex self-organizing phase-locked loop architecture, driven by bio-electromagnetic and electronic principles, minimizing free energy and optimizing energy transduction efficiency. Now, the real challenge we have in trying to characterize the function of this system is that the state of the system under observation is governed by fluctuations of many control variables, availability of oxygen, or not, food or pyruvate or not, Krebs cycle intermediates, NADH and succinate availability, ATP, ADP concentrations, relative pH, temperature, impinging light intensity and wavelength, and membrane potential, to name a non-exhaustive lists of the environmental perturbations and control variables that the system is subject to. However, it's argued here that the complex high-dimensional landscape of control variables at the microscale is integrated and trained and governed by a strong order parameter instantiated by the system. So I know I apologize if I'm teaching grandmothers to suck eggs here, but just a very quick journey through the schematic function of the phase lock loop. It is a predator-prey model Lotka-Volterra system, but with tight coupling between the input and output. A reference oscillator signal is compared with the output of a voltage control oscillator to generate phase signal based on the phase difference. That signal is passed through a low-pass loop filter. The output is a voltage which either drives the voltage-controlled oscillator's speed up or slowed down. So what we're looking for in trying to characterize the system is these components: a phase detector, loop filter, voltage-controlled oscillator, and the feedback path. What's missing here is that we also need to identify the reference oscillator in the system. So we're going to observe the individual components of the system, which form the key components of this, potentially form the key components of this phase lock loop architecture. So we're conjecturing here that the ATP synthase dimers form the basis of the voltage-controlled oscillator and demodulator, as we're calling it here. The cristae act as resonant cavities, waveguides, or Helmholtz resonators. The Mykos complex and the neck of the crista is an inductive gateway, and the electron transport chain, reference oscillator, and rectifier. And then we'll try to characterize the function of the whole dynamical control system governing the molecular behavior of this system. So we're looking here at an ATP synthase monomer. I'm not going to attempt to explain the detailed function of that, but generally speaking, these ATP synthase enzymes appear in the mammalian cristae around the rim in a V-configured dimer conformation, forming highly structured rows along the crystal rims, and we're suggesting that they function here as voltage-controlled oscillators. Their rotational speed is directly modulated by the proton motive force, ATP-ADP ratio, and electromagnetic, and we suggest here gyroscopic and vibrational forces to affect synchronized production of ATP. They act as demodulators, converting the high-frequency proton flux, a modulating wave of energy, into mechanical rotation that drives ATP synthesis. Under normal circumstances, the unidirectional high angular velocity rotation of the proton-laden C rings, which are the spinning components of the ATP synthase enzymes, generate significant vibrational, gyroscopic forces and distinct magnetic dipole moments, which we're suggesting in concert generate a strong order parameter. So difficult to visualize perhaps, but let's use some animations to see if we can do that. So we're really concentrating on what's called the FOC ring, which is the spinning turbine or pump or rotor, as it's been described. I'm trying to move us from that metaphor towards voltage-controlled oscillator. The spinning rotor in the center is called the FOC ring, O because it's been determined that oligomycin interferes with its rotation. So here we're looking at the stylized ATP synthase monomer, and we're looking from within the matrix, from within the very core of the mitochondrion. And here we see that proton flux, protons are entering into what's called the A subunit, which is the component of the system wrapped around the FOC ring. And in the mammalian FOC ring, there are ribs, 8 ribs there or thereabouts, each of which attaches a proton. the rim spins more or less through one complete rotation before, at the end of that rotation, a proton enters the matrix. So effectively, we're looking at a single coil solenoid with this system. So if we now move to look from the intercrystal space, from within the cristae invagination or flat sac, as I've described it, we see the same effect. Generally speaking, under normal operating conditions, the C-ring viewed from the matrix is spinning in an anti-clockwise direction. So from the other perspective, this perspective, it's spinning in the clockwise direction. And here we see the dimer conformation. And so therefore, with these horizontally opposed rotors effectively facing each other across the rim of the crista, they are contra-rotating and protons are flowing through them from within the intercrystal space. And here we're just removing the ATP synthase system and just showing the idealized, stylized proton rings spinning, contra-rotating, as if the ATP synthase dimer was invisible. So these are highly coupled. There may be weak electromagnetic forces in play here, but they're proton rings. They're spinning fast, around 100, 200 hertz, and they are closely aligned with maybe 15 nanometers between the rotors across the bottom of the rim. So if we then extend this, we see that the proton flux around the rim is highly organized, and that the individual rotors are very proximate, and they are, I posit, therefore very closely coupled and possibly synchronized. So here we see them, those proton rotors around the rim of the crista. And that provides the strong order parameter I suggest in this conjecture. So dynamic angled magnetic flux from the opposing and therefore contra-rotating ATP synthase C-rings create complex electromagnetic and hydrodynamic landscape within the confined crystallumen. We expect some kind of structured water gel within the crista. It's conjectured that these fields exert Lorentz forces on the protons flowing within the cavity. This results in coherent macroscopic rotational spin dynamics with the proton fluid, funneling protons efficiently towards the ATP synthase rotors and out of the crista, intercrystal invagination, further enhancing energy transfer. Nearest neighbor coupling and the mirrored conformation of the ATP synthase rotors, which are coupled chaotic oscillators around the rim, impart a strong synchronizing dynamic, as I've suggested. And proton egress from the cristae is a solenoidal, quite literally a solenoidal, in the electronic sense, component of the non-equilibrium steady state system function. So now we move to the MICOS, the mitochondrial contact site and cristae organizing system. We're looking at the side elevation here, or the front elevation of the mitochondrion, and we're seeing that the adjacent faces of the cristae. And then the MICOS appears as an annulus around the interconnection site at the top, connecting the crista to the intermembrane space. And there are very significant dynamics, proton and electron dynamics within this system. The MICOS complex structurally maintains the narrow crystal junctions, the necks, that serve as, I posit here, inductive choke points. Proton-electron flux through or around the constrictions generate significant self-inductance, providing dynamic inertia that stabilizes flow and resists sudden changes. The inductor the choke performs the function, I posit here, of the loop filter. It further couples dynamic interactions of the crista across opposing faces. And the tight proximity of adjacent crista, as I suggested, about 40 nanometer spacing between them, according to some research observations, means that there's potential coupling if there is a signal that propagates from within the crista across these adjacent components of the system. So if we try and observe the crista and consider its function and its physical properties, we know it's highly invaginated component of the inner membrane. It has very distinctive features, and we posit here that it acts as an electromagnetic resonant cavity, a Helmholtz resonator. I'm sure I don't need to say this, but the best example in everyday living of the Helmholtz resonator is the beer bottle we all blow across the top of, which generates a amplified sound wave at the neck of the bottle, which is a function of the resonant frequency of the air within the bottle. And the nanoscale structures we're looking at are tuned to specific frequencies supporting coherent proton and electron propagation within the confined lumen along their faces. Inspired here by a seminal paper by Mark Kag of 1966, Can you hear the shape of a drum? Well, it turns out that you cannot because there is more than one solution if you work backwards from the sound that the drum is capable of generating towards the physical structure that generates it. But given the structure, you can infer the eigenmodes and therefore the eigenfrequencies that the drum face or the crista face is capable of generating, or indeed can only generate. The permissible eigenmodes of the system therefore act to quantize the error signals that are generated by this system. And this provides the framework for the phase detector in the phase lock loop system. So here we're just looking at a very idealized, stylized view of the crista walls that are vibrating with the permissible eigenfrequencies adjacent to each other. And given the strong physical structure at the boundary of each of the faces, we can suggest that the Dirichlet boundary conditions are satisfied, so that we can indeed suggest that it is only possible for the faces to vibrate at the eigenfrequencies of the face. So just completing these two components of the system, the flat sac, like a deflated balloon, with the rigid structure of the ATP synthase dimer annulus around the top creates not just a boundary condition, but a symmetrical boundary condition across either face. As a consequence of that, we might expect to see wave patterns amplified through the MICOS. And I don't attempt here to describe the nature, the Fourier decomposition of that wave pattern, but suffice to say that the conjecture here suggests that the wave patterns that emanate from that intercrystal space propagate through the MICOS and across, as I said previously, to adjacent crista, where they will interfere with the individual stable operation of those adjacent crista to perturb them and therefore propagate signal across the entire intermembrane space. So now we introduce the electron transport chain. Electron transport chains generally are observed on the faces of the cristae, not at the rim. And we're going to just describe a little bit about the individual function of those components. This is a very stylized, radically simplified perspective, but we're looking at the four key components of the electron transport chain. The green elbowed electron transport chain is complex 1. The red is complex 2, the blue complex 3, and the yellow complex 4. The bottom of the slide is the matrix where the Krebs cycle is running, and the red dots are connoting electrons, electron flux. So the Krebs cycle delivers the Krebs cycle intermediates NADH to complex 1, rich electron source, and succinate to complex 2. And there is a cycling of reduction oxidation between complex 1, 2, and 3, which results in the generation of cytochrome C at complex 3, which propagates to complex 4. The net result of all of this, as you can see here, is that protons are pumped from complexes 1, 3, and 4 into the intercrystal space. And you can imagine the mirror image of this across towards the top of the slide occurring on the other face of the crystal. So complexes 1 and 2 of the electron transport chain are the only components of this system that are capable of oscillating in reverse, generating reverse electron transport. And it's conjectured here that this component of the system is the reference oscillator mitochondrial phase-lock loop. Complexes 3, 4, and 4 enable only bidirectional electron flux and therefore perform the function of rectifiers in a traditional electronic circuit. Complexes 1, 3, and 4 are cascading components of an operational amplifier transducer converting electron flux into proton motive force. And parallel or anti-parallel electron flux through the electron transport chain complexes, supercomplexes, generates resultant attractive or repulsive force, weak force, pushing the faces of the crystal together or apart, depending on whether the electron flux is in phase or in antiphase across the faces. In phase results in a force that pulls the faces together. So it's balanced at the same time, the system, by the proton pumping, because as the density of protons accumulating in the intercrystal space adjacent to the membrane surface increase to a certain threshold, then you'll start to see repulsive forces across the faces of the crystal. So you can imagine here that there's some kind of integral stability to this proton pumping electron flux system. But these are very weak forces, and the distance across the faces is in the order of 10 to 15 nanometers. So it is very small flux in relation to that distance we're looking at. So now we look at the ratio of electron transport chain complexes to ATP synthase enzymes within this idealized system. And by the way, observations have suggested that there may be as many as 100 of the individual ATP synthase monomers on each face of crystal. Here we're looking at around 50. So this is a sort of half-scale model of some of the cristae that have been observed in mammalian systems. And the ratio is the ratio of electron transport chain supercomplexes is in the order of 1 to 3. So 3 ATP synthase monomers to 1 supercomplex. This, by the way, is a highly stylized system schematic. They appear in perhaps a very much more random fashion across the faces of the crystal, but nevertheless, for the purposes of the conversation, perhaps useful. So that is the phase-lock loop system in essence, and we can now delve into perhaps some of the properties that imbue this system with a broad repertoire of what you might describe as computational functions. Here is the dynamic control system governing the molecular behavior. Just radically simplifies the kind of standing wave pattern you see in that diagram on the right-hand side is not the actual standing wave that's yet to be modeled. the forces that we've described imparted by the Micos, the electron transport chains, and the ATP synthase rotors, described as A, B, and C in this diagram, have not been modeled. But it is possible, I believe, to model the system given the geometry, given what is known about the extraordinarily well characterized stoichiometry and electrostatic properties of the system. Perhaps a little bit more risky, just looking at the system from the side, as we have with that stylized image we saw of the notional standing wave. We can expect the system to operate under normal circumstances, as I've suggested, with parallel electron transport on adjacent walls with coherent electron flux in the same directions. This minimizes energy loss, maximizes efficiency, and structurally stabilizes the cristae through attractive electromagnetic forces. As I've said, the reference oscillator, as described in this system, by complexes, by electron transport between complexes 1 and 2, can under certain circumstances reverse. This performs a function of so if it switches to reverse electron transport, which it does under high proton motive force conditions and reduced, high concentrations of reduced ubiquinone, or ubiquinol rather, complex 1 flips and becomes a proton pump rather than a proton consumer as a function of this reverse electron transport chain. That's, relatively speaking, violent transition. But if we look at the mirrored architecture across the opposing faces of the crista, then all of a sudden, at least I can see a physical and an electrostatic and electrodynamic structure that is very reminiscent of, analogous to, perhaps even is, some kind of complementary transistor amplifier flip-flop. And if it is, that's, as we all know, the foundation for the modern computing industry. So, now, even more controversial, perhaps, but I think towards the active inference community, open to the strongest scrutiny. The rotating C rings of the ATP synthase enzymes will generate gyroscopic forces. Magnetic dipole moments generated by the C rings and the electron transport chains will interact with Earth's ambient magnetic and gravitational fields, which will act across the entire organelle. We're saying here that the mitochondrions' spatial attitude, orientation relative to these external fields, as it changes, will induce subtle precessional torques on the ATP synthase rotors, which are gyroscopes effectively, and alter the coupling electromagnetic and gravitational with the external fields. These induced changes could feed back as detectable modulations of ATP synthase rotation, proton motive force, flux, or electron flow, allowing the organelle to, dare I say, sense its spatial orientation and changes therein. This might be perhaps a fundamental mechanism for cellular proprioception. Indeed, it's not too big a stretch to conceive of that as more than just a metaphor. I happened to work 40 years ago on inertial navigation systems before GPS rendered these systems redundant. And these very simple coupled oscillator arrays, 3 gyroscopes, some accelerometers, and some strain gauges were capable of operating as an autopilot to get an aeroplane from Heathrow across to JFK with only meter errors on landing at JFK as far back as the Second World War, in fact. So I want to just briefly touch on a couple oscillations with the Krebs cycle. I've mentioned that the Krebs cycle is operating within the matrix, the cycle of life. The Krebs cycle introduces Krebs cycle intermediates that are available to the oscillatory system that I've described within the cristae. And there is a tight coupling of the feedback loop related to ATP, ADP concentrations, NADH, NADH plus ratios. I don't, and I haven't attempted to explain the extraordinary process that operates within the matrix, the Krebs cycle, in the context of the system model I'm building here. But suffice to say, it's been observed in bacterial organisms, E. coli, that some protein systems are generating standing wave patterns, and the standing wave mode is a function of ATP, ADP binding and dissociation rates. So at the the rims of the ATP synthase dimers that we're looking at here, the mushrooms, as they were called when they were first observed under microscope, ADP and inorganic phosphate are being transformed into ATP. The reverse process in the bacterial organism and the signal that binding and dissociation creates has been observed to generate standing waves. The obligatory self-organizing criticality slide in any dynamical system presentation. I don't want to insult you by taking you through these individual components, but the question is, what is the order parameter and what is the control parameter, or what are the control parameters that position the mitochondrial membrane at the critical point so that it's most sensitive to these external perturbations, the control variables, and propagates the signal with maximum fidelity over the longest distance across its entire membrane and to adjacent mitochondria. And my suggestion is that the control parameter, the order parameter rather, as I've said, is a function of the strongly coupled array of oscillators around the rim of the crista. And the control parameter is an integration transduced and propagating through these vibrational modes onto the membrane, perhaps. So I just want to move towards the bio-inspired computational direction, which is really where my interest lies, beyond the characterization of the system that I've attempted here. And I just want to reference the works of two of my collaborators, Dr. Tjeerd Olde-Scheper, who is at Oxford Brookes in charge of the robotics lab there, and Dr. Yoshikatsu Hayashi, who's at Reading University, a biophysics professor there. And their works in relation to rate control of chaos, in the case of Tjeerd, and anticipatory synchronization in the case of Yoshi. So Tjeerd has done work to describe a control function that can be imparted to a coupled oscillatory network such that it is constantly positioned at the boundary of chaos, but across at the critical point, or slightly hot, supercritical, or slightly cold, subcritical, but nevertheless never entering the chaotic mode of the phase plane. And then Dr. Hayashi's work is very exciting as well. He's developed a system that describes how a coupled oscillator array with appropriate delayed feedback mechanisms can be utilized as the entrained enslaved component of a system. It can actually be influenced such that it starts to anticipate the chaotic behavior of the master in the system. So I find this extraordinarily exciting, that potentially here we're looking at a system that could exhibit anticipatory synchronization. So just want to very quickly say that these phenomena are going to be incredibly difficult to sense. I do believe it's going to be possible. We're at the limits, and really I'm hoping Carl can give me some guidance on this. We're at the limits of the sensing technologies that will enable us to observe these oscillatory phenomena on the membranes of individual mitochondria. But I'm nevertheless talking to instrument manufacturers and experts, one of whom is a collaborator I'll mention at the end, towards trying to figure out what sensing modalities could be utilized to observe this flux, these wave patterns on the mitochondrial membrane. But here I'm just showing a couple of images that show the alignment of cristae invaginations or flat sacs across adjacent mitochondria. And the question I pose here is simply, could the phenomena that we've described here, in some way, shape, or form be responsible for fission and fusion? And might the kind of system that we're describing here form the basis, at least for one mode of communication between mitochondria and between the networks of mitochondria and the host cell? So just moving further towards the bio-inspired computational paradigm, what we're looking at here in this Huckan-inspired synergetic system with complex control parameters, nevertheless integrated such that the system is radically simplified with this order parameter I'm suggesting. The system provides a clock function, and it's the same standard clock architecture that's used in more or less every classical von Neumann compute architecture today. The system we're describing here is analogous, but subtly different. And I don't plan to go into it here, but one of the scientists who has greatly influenced and inspired me throughout my life, Gordon Pask, originally described by Heinz von Forster as the cybernetician, described the requirement for the coherent clock function across adjacent actors in his interaction of actors framework as the fundamental basis for a conversation in accordance with his conversation theory. What we're looking at here is not a clock that doesn't change, but a clock that changes based on perturbations to the system, but also enables perhaps clock coherence across adjacent organelles. So I want to also say I had the great privilege of having a conversation with Ricard Solé recently, and he alerted me to his extraordinary work on the liquid brain, an ant colony as liquid brain, perhaps a microbiome as liquid brain. The suggestion here is the highly dynamic network of mitochondria within an individual eukaryotic cell might also be a liquid brain. So I won't dwell on this slide. Apologies for the poor image, Carl, but perhaps we're looking at a minimum observable active inference engine in this structure on the mitochondrial inner membrane. So I think just looking at what's happening in the current AI landscape, we have perhaps three significant vectors along which people are suggesting the path towards a more sophisticated cognizing systems might lie. One is the Google primary mode, which is large language model with deep reinforcement learning. Jan LeCun's hierarchical joint embedding, predictive architecture, and of course, Carl et al.'s work on renormalizing generative models for scale-free active inference. I believe that the inclusion of models emulating this mitochondrial function might present a compelling avenue to explore towards scale-free active inference along the trajectory that Carl and the community are pursuing. So the broader significance of this, I believe, is potentially exciting. It might reshape our understanding of communication and control in mitochondria and networks of mitochondria and their broader function within the cell and the multicellular organism. The scientific and commercial implications, I think in the short term, are exciting. perhaps slower burn, but novel therapeutic strategies based on this different understanding, breakthroughs in cellular control, health span and lifespan, given we're looking at a fundamental clock structure here. But I'm particularly interested in the next-gen technologies, low-power computation and bio-inspired AI, adaptive biomimetic sensors, and proprioception mechanisms for future autonomous system design. So that's what I wanted to say today. I want to give great thanks to the people that I have engaged with and had wonderful conversations with and who've been supportive over the course of the last year. The number of conversations I had with Thomas Parr, who has encouraged me on the way, have really inspired me. The collaborators, Yoshi, whom I mentioned, Munehiro Asai, who is focused on biophysics of bacterial organisms, tiered control systems and rate control of chaos. Sven Zewitz, a former colleague, now with Frontier Strategy, a biologist. Bo Ivanov at the Cambridge Mitochondrial Biology Unit. Sid Ahmed at Nottingham University, focused on ion sensing microscopy. And Dr. Yamuna Krishnan, University of Chicago, who is one of the world-leading chemists, having created a technology that can sense membrane potential across many of the organelles within the eukaryotic cell. Andrew Acorn, who helped me tremendously with these stylized animations. Radwan Udriri, a long-term colleague and cybernetician and control theorist, as well as the Guy Foundation for Quantum Biology, where I first tested these ideas more or less a year ago and got the enthusiastic response, which encouraged me to evolve them rather than abandon them, which was my intent at the time. And finally, the encouragement from Tim Verbellen, who I had a conversation with and who encouraged me to participate and deliver the presentation today. So thank you.
Thank you so much, Anthony, for the presentation. Really cool. So now we move to the questions and comments, if there are any. I just have one general about, so what are your thoughts, or how can it be more related to like the neuromorphic side of hardware, like in machine learning in general? Because, you know, like certain biologically plausible algorithms like predictive coding, like are like severely suffer from standard GPUs because it's really hard to do like speed up inference in those types of hardware and so on. So what can be like kind of directions, or what are your thoughts on that?
Yeah, Miguel, thanks. I haven't applied my thinking long and hard enough to that question, but I'm interested in membrane computing, reservoir computing, echo state computing. So I'm very much inspired by the neuromorphic landscape. But I think what I've been particularly interested to do is try and further characterize the biological computational repertoire and by extension cognition capabilities to kind of expand the view. I think, given that mitochondria are imbued with these extraordinary structures, and by the way, those structures are not in evidence in the common ancestor of the mitochondrion. So it's a very specialized system geometry that's evolved within the higher order living organisms. So I know that if I go hard towards the crazy end of the spectrum, the current paradigm suggests that the ATP, the mitochondrion generates the ATP. It's the battery. If you go to the other end of the spectrum, you could say that it is highly conserved, interlinked, but autonomous community within an individual cell and across a biological, multicellular organism. It retains its autonomy, and so until such time as we understand the interaction of mitochondrial networks within an individual eukaryote or across a multicellular organism, and you know, given the scale we're looking at here, there are quite literally hundreds of trillions of mitochondria within an individual human. So until we kind of can reshape our perspective, the lens through which we view this, then I think the neuromorphic landscape is going to be missing, you know, part of the picture, if that makes sense. So I very much would like to, you know, having been a technologist, you know, control engineer, and you know, having built software companies over 2 decades, I very much would like to now move towards exploring that landscape and trying to figure it all out. But I've been very much focused on trying to understand how, you know, or rather expand the perspective and the view of what biological computation might actually look like prior to doing that.
Okay, yeah, makes sense. Cool. Thank you. Yeah, Andrews, I have a question there.
Yes, Anthony, thank you for your talk. So informative. I learned so much.
Thanks,
and I only learned a small part of what you said. But so first, I have several questions. So maybe I'll let others speak too. But clarification question. When you say opposite faces of crista membrane, are you talking about a single crista, or are you talking about?
Well, so I've been very careful not to try and explain or understand beyond what's already been characterized, what's happening within the matrix of the mitochondrion, other than the brief allusion to it in the presentation as providing the Krebs cycle intermediates that couple the matrix Krebs cycle oscillatory system with the crista phase lock loop structure. So I'm really talking about adjacent faces of an individual crista in this conversation. That said, as I mentioned, the individual crista monomers at the rim are in close proximity to adjacent crista along the mitochondrion. You know, some estimates as little as 40 nanometers separation between the two. So there is coupling, not just I believe on the membrane, but there's coupling, strong coupling across adjacent crista within the matrix. I don't know how it's going to be possible to characterize that.
So the primary relationship is within its own cavity, and the standing waves are on the boundary, or are they going all the way through? How do you?
So we have this complex proton flux dynamics on the rim within the crista, intercrystal space, within the invagination space. We have the supercomplexes of the electron transport chain, you know, in the ratio 1 to 3 on the individual faces, the opposing faces of the crista, imparting electrodynamical modulation of the
within the
individual
crista,
faces, but in
within the
in
crista,
accordance with the eigenmodes. And then, as I say, at the top of the system, you have the mitochondrial interconnection site and crystal organizing system, which I'm conceiving of in this model system, this notional model system, as a choke in the traditional electronic sense. So an inductor capacitor. And you know, that will seek to minimize flux across the faces to the extent that that flux, transitory flux, you know, is in evidence. So it acts as a low pass filter to reduce the perturbations such that coherent same directional electron flux is occurring on the adjacent faces of an individual crista. That's my current conception.
And then to ask another, thank you, that's very helpful. And then to ask another question, when you talked about the sensitivity of all these things, I think for the Krebs cycle, it has to worry about. And then when I think of the 8 steps in the Krebs cycle, and I've written you, like, I've been studying Both periodicity as a it's a fantastic structure in mathematics that is basically cycling through the symmetries of mathematics, because it cycles through the way that the classical Lie groups, orthogonal, unitary, symplectic, are all embedded each other repeatedly, just on and on and on. And so because I'm a metaphysician, I look at philosophy, so I'm introspecting things like you had on your chart, something like a learning cycle, like you take a stand, follow through, reflect. So or like, free will versus fate, or whether, what, how, why, levels of knowledge. So imagine going, the system, let's say, going through such things. And these are the contexts that allow you to align up, like what we have in active inference, where you have two probability distributions. So imagine a dialogue between a probability distribution that is direct access to evidence and a much more compressed one that is simply inferred causes, right? They're trying to communicate. So it's like in quantum foundations. You have answers, states are answers, but you have observables or questions. You have measurements that relate them. And it turns out quantum symmetries, there's 3 quantum symmetries. There's time reversal, that would be for the answers. But charge conjugation says, well, what if you flip particles and holes, or particles and antiparticles? You see, then it's kind of like questions, like flipping answers and questions. And then you have the combination, which would be mirror symmetry, saying like, well, a left counterclockwise cycle is the same as a right clockwise cycle. So that speaks, all these things are very metaphysical. So imagine if we were in that Krebs cycle, and what it's doing at each stage, it's like an antenna. It's saying, look, do I have the precursors I need to go forth? Because if I can't, I won't, you see. And so it's saying, now this is the prevailing context, that this is where I'm stuck, right? And what does that mean? What does it do? But it's certainly like an antenna for a particular problem. And it has like 8 problems or 8 contexts that it has to cycle through. And that's the life of the Krebs cycle. So like for a human being, it would be like, you start with God, and then you have order, and then existence, and then participation, and knowledge, and decision-making, morality, logic, and back to God. So that's a human being. But let's say a mitochondria just is saying, well, I need, carbon dioxide, or I need oxygen, or whatever I need, right? It's cycling through. So that's the picture. And the Bott periodicity says that you can get all those combinations by cycling through combinations of those quantum symmetries, because they get this for topological instances. Like if you look at a Fermi on an electron all by itself, that's what its life looks like. So I guess maybe the question is it possible that the Krebs cycle is playing the role of that type of antenna for the relevant context?
Yes, well, that's whether it's a Krebs cycle or not. It's difficult for me to conjecture further. But Andreas, we exchanged a few notes prior to the call, and as I've suggested to you, my mathematics is rudimentary. Undergraduate, third year undergraduate, alongside my cybernetics, but nevertheless rudimentary. But of late, over the last year or so, I've been reading up on the topology to try and understand the geometry and topology of this system, and how it might transduce perturbations and transpose those perturbations from one coordinate system to another coordinate system. I'm still not talking about the Krebs cycle, but I know that in a lot of the work in explaining visual transduction, we're using Fourier analysis and Gabor wavelet analysis, and I've been very much excited by the application of approaches to understand and explain that, because I think we can describe edge detection here as some kind of fundamental computational primitive that the system is capable of performing. So if you like, also there's a group or a category or categories or groups of perturbations, as I've described these control variables that form groups and classes, and at the other end of the spectrum, or rather at the output of the system I'm describing, is a Fourier decomposable wave pattern. So we're looking at archetypal transduction from edge detection, ramp, spike, step function in any one of those control variables towards a wave pattern, which is the output of the system. So I think that it might be possible to describe the geometric, topological, as well as the kind of mechanisms that I've sought to explain here today, to characterize how that transformation from one coordinate system to the other is performed. the question that you ask, I mean, we're looking at magnetic dipole moments. I mean, I said that the proton flux from the intercrystal space out into the matrix is quite literally a single coil solenoid. You can use the solenoid rule to describe how, given the direction of rotation, where the north-south poles will lie, and then you can imagine, you could also characterize this whole system as a phased array antenna, and so there will be mechanisms to describe the character and the performance of that phased array. For instance, given that if you suggest and try to characterize that ATP synthase electromagnetic system around the rim as a phased array, then to the extent possibly that any individual oscillator in that array is no longer capable of maintaining its state enslaved along with its nearest neighbors and the collective, in submitting to this order parameter, then that will result in some kind of directional propagated signal from the failed oscillator. If I just take the work I did 40 years ago, maintaining the weather radar on the Concorde, have a flat plane phased array. In fact, modulating the individual slits in the phased array was how one achieved the directionality. So by direct analogy, you could understand how this antenna array would generate sort of directed field in the event of any kind of individual failure. But to your point about broader topological mechanisms, Lie groups, etc., I emphatically believe that they will be applicable approaches to describe the transduction from the one coordinate system to the other, given the work that's been done in the visual field, and most recently, I think in the explanations of the two-dimensional Fourier-Gabor patterns that we're seeing in the initial layers of large language models that have stabilized around a learning function. The same approaches, I think, can be applied here notionally in building the system model. Whether we can observe them or not, question mark. But the challenge, of course, we have here is that we're dealing with a biological system that is evolving in time, and it might appear, at this scale, observer relative to be symmetrical in some way, shape, or form. But we know that it is a biological system, and therefore we're dealing with broken symmetries. So, as I suggested to you, I've been very excited about recent publication employing Grothendieck's symmetry fibrations to try and explain how message passing can be affected in biological systems to generate local synchronization within components of an extended network using the groupoid mechanisms.
It's very fascinating. I have one last question, but I would ask. I don't know if there's, if I may, but
yeah, go ahead.
Yeah, and so the point is, well, and so you're talking about spatial, and that would be fascinating, if you can do that. I think from the point of view of mitochondria empathizing with one, that their world is chemical. Maybe I'm wrong, but they have because I'm into the Krebs cycle. There's more to it than the Krebs cycle, let's say. But a crucial thing with the quantum symmetries is that they can square to +1 or they can square to -1. And you talk about the phase, and I'm wondering, it's kind of like an essential feature. And I'm wondering if that distinction may pop up somewhere. And that notion of like holes versus particles. So in a sense, you alluded to that with like the broken array, which can actually function, have a positive function in a certain sense, but that there's other things where, like if you flip the direction of the cycle, or if you, where in one direction, it may be the particle direction, but in the opposite direction, it's the whole direction. Let's say something like that, where you have two languages, you have two ways of looking at things, and so you have two different kinds of bases for symmetry, and it's anyways, that's what I would be looking for, I think.
What I'd say to that, Andreas, is that I suggested that perhaps the electron transport chain is a cascading operational amplifier, or because it's electron flux in the reference oscillator and proton flux in the output, it's not strictly an amplifier, it's a transducer. But the characterization that I've sought to deliver here today is in no way, shape, or form sufficient to really describe proton pumping and electron flux. Of course, when you think about the electromagnetic components, or rather the conductance, resistance, inductance, capacitive properties of these individual metalloproteins that constitute the individual complexes, you can't do that without understanding electron hole dynamics. But I'm not seeking here to characterize a quantum model. And when I talk about quantization,
I'm not. But that distinction, for example, between protons and electrons, that you have two different types of conceptual objects, and then all of a sudden it's a richer game. So I think, so I'm not thinking about quantum physics here directly, but just by analogy, so thank you, and I'll certainly contact you afterwards. I appreciate your talk very much. Thank you.
Thank you very much.
Thank you so much for the questions. So I guess we can now move to Eukaryotic.
Thank you. Is it my turn now?
Yeah, it seems so.
Good. Well, thank you, Anthony. That was an encyclopedic and enlightening presentation. I don't think I've ever met anybody who has such an intuitive, formal understanding of the mitochondrion, and it's the implications of that. So just to qualify my summary and response, I'm probably worse than you in terms of the maths. I did do physics at university, but I've forgotten it all. And as you say, apart from the endearing arrogance of physicists, that's the one thing that I have retained. But I've forgotten all so I have to go to Wikipedia whenever writing an equation. Just to frame the response and how we or I can usefully feedback. How did Thomas Parr and Tim Verberlin suggest that it may be useful for you to present here? What did they say? Oh, you should go and tell Carl about this, or perhaps you could ask Carl if this or that.
No, so Thomas, I've been speaking to. I met Thomas at Active Inference conference in Leiden at the beginning of last year. Had some very interesting conversations with him. He's very interested in the sort of the fundamental thermodynamical basis to describe active inference in any system. So I just described to him that my interest in mitochondria and my system model. He talked about. I should say, Carl, that so one of the, if you like, stimuli and the trigger for this was a conversation between Nick Lane and Mike Levin a year ago, where Mike Levin asked the question, could we build a model of the mitochondrion? So that was kind of spur for my initial interest. And I started to describe that in my initial conversation with Thomas. I mentioned that I was, working with the Gaia Foundation for Quantum Biology, and that I'd like to introduce him. So we had some good conversations, but really it's been me seeking to, with a cybernetics background, step across from, my world into his and your world in active inference. I've become very excited and enthusiastic about the approach, generally speaking. But then he talked to me about, work he was doing with Anil Seth and referencing Nick Lane's work and Mike Levin's, and it just seems that there's a there's a rich epistemic landscape where there's an intersection. So I've just been talking extensively to him, and then I had a cause to speak with Tim, and he suggested that I perhaps give a talk here. And then I mentioned that to Thomas, and he very kindly helped me prepare with the first draft of the paper. But he's so busy, as you know, that he hasn't had the time to talk after that. But I very much hope to continue the conversation with him towards trying to bridge this world that I kind of inhabit with my frame and picture, and the kind of the direction that the field and what's next in artificial intelligence will take. So I just wanted to be part of the community. There wasn't any specific impetus other than Tim's suggestion that I put my name down, approach Riddy, and that he would give me the thumbs up in the background. That's how this came about.
Right, good, excellent. And so the work that Thomas is doing with Anil, Seth, and Mike, does that involve Nick Lane, or is that separate work?
It doesn't, as far as I'm aware. I about a year ago, I put a very clumsy, rudimentary paper together outlining my ideas and sent it by email to Nick and to Mike. Both both responded to my original outreach saying, send me something, send us something, and we'll seek to comment. But I haven't had any interaction since. Interestingly enough, both Nick and Mike, some of their work has been supported by the Guy Foundation, which is how I originally came across the Guy Foundation. I've been following both of them for over 10 years. And so that was exciting. But I haven't engaged directly with Nick or Mike around these ideas, and I don't believe Thomas is talking to them directly. Although Thomas did send me a draft of a paper he had, a commentary he'd been asked to put together on a paper by Mike Levin and Chris Fields on some of their work. That's kind of the landscape, if you like.
Right. No, that's very useful. Yeah. So yeah. Interestingly, that was going to be the sort of the major portion of my feedback. You need to be integrated into the right community, and that integration could be at a number of levels. So I'll ask you another question. You have this, I repeat, encyclopedic knowledge, which is formal, deeply informed by cybernetics and your memory of high school maths. And you could use that in one of two ways. You could use that as the basis of a forward model, a complex system model, a phase reduced model, a mean field approximation, to understand mitochondrial function using data from mitochondria. So basically getting into the game of complex system modeling in a formal and informed way that would enable you to make sense of empirical data in relation to the dynamics and the principles of self-organization that you could bring to the table. And that could have implications for therapeutic interventions. It could have implications for theoretical biology generally before you translate into a clinical or a therapeutic context. So that would be one application. And at that level, you wouldn't really need to refer to active inference or indeed artificial intelligence. So there's another angle, which is, well, if I can understand this kind of self-organization at this nanoscale, at this mitochondrial scale, can I now use this, leverage that understanding to motivate a neuromorphic approach to artificial intelligence and machine learning? So that would be, if you like, a reverse direction back from the neurobiology or the biology in this instance to the engineering, as opposed to using sort of formal engineering or mathematical constructs to to inform our understanding of this sort of microscopic biological self-organization. I think you have a choice there, because who you talk to and where you invest your time is going to be dictated by what you commit to. So if you had the choice, which one would you commit to?
Maybe a little bit more context, Carl, because I'm in spirit and just as a function of training, entrepreneurial. So I'm interested to create something that utilizes this perhaps unique perspective that I have. So the question is how and what, and I have until now resisted, contemplating starting a new company, building a new company. But if I were to do that, it would be towards the neuromorphic meets active inference, direction, because instinctually I know that that's what's next, and I find that very exciting. However, I've got experience over 20 years of what it is to be building a venture-backed business where you are rewarded for exploitation rather than exploration. And of course, within the academic realm, few people achieve your prolific path in defining a new field, because then the incentives don't work there for sort of moonshot innovation. But in the middle is this space which is increasingly viewed as interesting by some of the leading entrepreneurs funding research around the world towards a dedicated research endeavor or a focused research organization. If I were to pursue the path towards some kind of therapeutic or some kind of transformation of the health system based on these ideas, it might take me 20, 30 years. It might take, the best case 10 years. So I think the most proximate opportunity is towards the neuromorphic stroke active inference computational organization in the next phase. But I've been seeking to attract, and I only really started the journey this summer, to attract philanthropic funds to create some kind of dedicated cross-disciplinary, cross-organizational research endeavor where we can incubate the ideas, evolve the system with the experts across the domains such that we get more confidence in it, so that we can seed the health and therapeutic opportunity in the future, but perhaps in parallel I can start to contemplate building the company. So I submitted an application for the Renaissance Philanthropy Big If True program, which they launched this summer. Renaissance Philanthropy is closely aligned, associated with ARIA, the Advanced Research Invention Agency. And whilst not successful, I got really encouraging feedback from them saying we would encourage you to continue to de-risk this endeavor and move towards a dedicated or focused research organization and program of work. And that program of work might span a 5-year period. And I have certainly the appetite for that. But I also have the appetite to bring together the community of interdisciplinary thinkers that are energized, and indeed I already have been quite successful in attracting and pulling together that community, and I'm doing exciting work with them. Now, I do believe that this is I'm not just sort of shoehorning the active inference framework into what it is I'm doing. I think it's fundamental to the kind of direction of travel I want to take in that neuromorphic computing direction. And so what I'm hoping for is some kind of alignment with the active inference community, collaboration with the active inference community towards evolving the model, because I do believe emphatically that will bring that will be an attractor function in interesting the philanthropic community that I need to crowd in around this endeavor.
Okay, that's very good. Okay, so I'm just thinking practically, how's that, making that work for you and you making it work for everybody else who you might want to engage. So one sort of practical way to leverage what the investment so far, intellectual and otherwise, would be, for example, say that I am well positioned to get philanthropic funding for an institute without walls or some organization that may have the resources to support, say, a postdoctoral fellow or a couple of PhD students that
Yes.
are supervised by the good and great, both in the theoretical side, could include active inference or indeed large language models, right through to experts in empirical mitochondrial function, such as Nick Lane and possibly Mike Levin. And that to explore such an initiative, I am going to organize an online workshop in which I can put forward this idea and invite all the great and good just to see if there's any interest out there. The purpose of this workshop is to scope the space for interdisciplinary collaboration, focusing on generative models of mitochondrial function as a canonical, if small scale, example of biotic self-organization and computation, basal cognition of a certain kind. The ultimate agenda in 5 to 10 years would be to take these principles once validated against empirical insight and deploy them in artificial intelligence or generalized artificial intelligence research that might touch on engineering and machine learning. Would that be the kind of thing you're looking at?
That is the dream outcome that I had in mind coming into this conversation, Carl.
Okay. Right. Okay, that's good. So I know what I will do now then is while you were talking, I was just thinking, how can one substantiate, help, sort of put leads out there that follow up on some of the directions of travel that you mentioned, by picking out some papers, usually the papers that we've been involved with, but also just the names of the players. I think you would be, it would be good to engage at least in this sort of fictive, let's get together on Zoom and talk about how we could help or contribute to some virtual institute that you're seeking funding for. And I'll briefly.
By the way, Carl, just very briefly, you just reminded me that was Thomas's suggestion also. Could we put together some kind of Santa Fe Institute style online conference whereby people could share their work and then converge to the extent there was a convergence as a function of that?
Yeah, excellent. Okay, so you may put, although he's very busy,
yes,
probably not as busy as I am, but he's still exceedingly busy. You can make him do some of the work. Okay, so that's good. I mean, I have to say, I should qualify this feedback. You kept saying, I'm sure you know about this. Thomas is a neurologist. I'm a psychiatrist. The last time I heard about a mitochondria was 4 decades ago at school. So a lot of this is double Dutch to me, as it is in my conversations with Nick Lane and Mike Levin. But I watch on admiringly. But I do. So but the kind of feedback I can give you is going to be very sort of conceptual and just say, have you thought about looking at this basic phenomena from this perspective, and could you use that as a selling point to a funder, for example, of your philanthropic institute at Santa Fe? And there were certainly a number of points during your presentation that say, oh yes, it's got to be like that because this is how self-organization works, and this has been leveraged or exploited in this paper. So I'm apologizing in advance that the links between what you presented and what I'm briefly going to rehearse for you are tenuous, but 1 or 2 of them may be useful when writing down the motivation why I've invited this person to talk about models of the mitochondrion.
Super.
Let me grab the screen and then see that I remind myself what I was thinking. Sure. I'll talk quickly because I'm aware this is being recorded and you must be getting tired now.
Oh, not at all.
Right. Well, I'll still talk quickly.
I don't want to take up any more of your time than I must, though, because I know how busy you are.
Yeah. Right. Yes. So Yeah, in no particular order, I think. So I'm just going to remind myself by clicking through these things. So I think this, in terms of the the overall change in direction of travel of in machine learning and artificial intelligence research to neuromorphic or at least biomimetic computing, I think that's absolutely right. And you could even try and sell that just before the sort of the the current generative AI bubble bursts. One may have to wait until Trump is removed, because at the moment all the money is on going in exactly the other direction, away from biomimetic and self-sustaining um, principles you might want to find in terms of a sort of universal intelligence or natural intelligence. I just put this paper up there as a nice little survey. I'm a co-author, but I hardly wrote any of this. This is basically Alex's contribution. So it's a really nice synthesis of different perspectives on mortal computation. I think that may be a nice catchphrase, and I'm thinking here again, when you go back to your funders, saying, can you pay for a little workshop in order to scope the possibility of polishing this and de-risking it and getting some commitment from people in the field. If you sell the mortal computation angle, I think that's quite neat, because what you're talking about at the moment is the brutal inefficiency and the danger to the climate of immortal computation, where immortal computation is just software running on anything, and of course anything includes sort of supercomputers with nuclear power stations to keep them going. But once you go,
a catastrophe.
Absolutely. So I think there's really, there's real pressure on all of us to move to viable mortal computation, where essentially that's substrate dependent. It's actually getting into the weeds in terms of the self-organization that has this aspect of computation under the hood, and that can be framed in terms of memristors. There's some interesting progress that I was being told about last week from Extropic, has just sort of gone non-stealth mode. If you wanted to, I haven't done it myself, but you look at Extropic in America, they're basically developing chips where
these are the thermodynamic
absolutely
chips. Yes, I have looked at them. Yes, I have.
Yeah. So I mean, that's not got the phase reduction or the Fourier aspects that you like, which of course it should have, and we'll come back to that later. But at least it's a move to actually substrate dependent true mortal computation. But you could also think about memristors, photonics, and all that good stuff before you even get to neuromorphic. So I thought this was an interesting discussion from a number of perspectives, but and I'm just putting it up as a nice synthesis. I'm hoping, if I remember correctly, that it is fairly exhaustive in its review of different perspectives on mortal computation. It doesn't sell why it's important, but it does at least put it here as a, sort of a counterpoint of reference to immortal computation.
Yes, Carl, I can't see it. Have you shared it? Can't see it.
Yes, no, I'm sharing. I'm assuming that. Have you stopped sharing?
Yes, I've stopped sharing now.
Yeah, I can see what you're sharing, Carl.
Miguel, perhaps you could just I could connect with you afterwards and you can forward it to me. For some strange reason, I can't see it.
What are you seeing on your screen?
I'm seeing you, Andreas, and myself on my screen. I'm not seeing anything in the chat or anything.
Andreas, what can you see?
I see the shared screen, mortal computation, and I see all of you.
Right, on the side, presumably.
Yes.
Yeah. Am I moving at the moment?
Yes, you're waving.
Oh, that's very strange.
I don't know what it might be, but I was explaining to Miguel that just minutes before the call, the broadband went down in my adjacent office, and so I had to move back into my home, and I don't know whether that's I doubt it, but there's something going on.
Do you want to try and sort of go out and come back in again and see what happens?
Yes, by all means. See you in a moment.
Okay.
I just want to thank you for hosting these theoretical neurobiology meetings. It's really a great privilege to get to participate. Thank you.
Thank you. Yes, I don't know how long we've been doing them now. We've only had the chairs for Miguel, how long have you been a chair?
I think it's been a year and a half, maybe already, something
Right.
like that. Yeah.
Yeah.
Oh, I think Anthony's back.
Thank you. Yes, I see that now.
You're right. Okay, let's hope it changes and your computer. So yeah, that's the paper, Mortal Computation. I think it must be published somewhere, but this is the archived version. So that's just
Okay.
a point of reference. You've mentioned Chris Field's, and I just put this paper up as again just something to nod to in any proposal, in the sense that not only is the neuromorphic angle the clear way forward in terms of artificial intelligence research and computation, computer science generally, but it's almost prescribed when one takes a sort of quantum information theoretic perspective on
Yeah.
certainly under the free energy principle. So, yeah, I'm not saying you go quantum in the same spirit that Andreas was advising, is disclaiming, but Chris Fields might actually want you to. But it's just a nod to, there are principled reasons, certainly under the free energy, the quantum information theoretic formulation of the free energy principle, that you might want to go to sort of neuromorphic and tensor, sort of a tensor neural network kind of implementation.
Yeah.
It doesn't have the solenoidal or sort of oscillatory or the aspects that you would, I think, want to foreground, but it's another nod to the ubiquity of neuromorphic principles. A lot of this will be redundant, because what I was going to suggest was you really do need to engage Nick Lane and Mike Levin on
Yeah.
that side of things, or at least have their titular support to move this forward. Simply because there are so many joint agendas in terms of taking biotic self-organization in the direction of engineering and making it work for the greater good, that are aligned with Mike Levin's insistence of sort of destroying neuro-chauvinism and just saying there's computation at every level, everywhere,
Absolutely.
from the mitochondria through to societal self-organization. And yeah, I just put this as one of 100 papers that he's written recently. And of course, Mike is not only a friend of Nick, but also a friend of Chris. So this is a little cabal you need to engage, I think.
Yes.
And I'm sure you've read that. Where is Nick? And I'm glad that you've. So I didn't realize you. Are you on speaking terms with? Not speaking. Not on a friendly
No.
terms.
I've asked some questions of him, online at Gaia Foundation events where he's presented. But no, I've had similar interactions with Mike Levin online, but I'm not yet acquainted with either of them, in a particularly direct way.
Right. I think you need to be, and it's a question of how you engineer that, which is why I was
Yeah.
this notion of having an online session that they're incentivized to join because there might be funding for their students. That's basically
Got it.
the agenda.
I think I can engineer that.
Excellent. And just to give you a bit more power to your elbow, I myself, Nick, and Mike are currently founded, I think, by the the Bill Gates Foundation. So we have an ongoing project which is now in its second year, looking at mitochondrial function.
Super.
And the idea was that I was going to come in as a sort of the theoretician looking at the biophysics, and in particular helping model the data that are currently being acquired by I think it's Eugene and Nick Lane at UCL, and I'll just put these names on you know on the recording so that hopefully you'll be able to speak to these people
Amazing.
in the time. Franz Kutschling and Axel, who are working at Tufts under Mike. And their job is basically, well, actually Franz is earnestly collecting data, but certainly Axel's job is to come up with complex system models cast in terms of kinetics and stochastic differential equations that will rest very much upon the kind of phase reduction or mean field approximations that you have been engineering over the past few years. To not just to understand the system and look at its see how the dynamics inherit from the functional form, but to actually fit the control parameters to experimental data. And so that part of the challenge is yet to be met. But there is a forum which you might want to become involved with initially, possibly as a spectator, but later possibly as saying, well, look, hang on, I've got a model of this. Do you want to think about this particular model? There is a forum called the DCM Work Group that is organized by Johan Medrano, who is currently at MIT. And this meets on Wednesdays at 4:30 for an hour and a half, and has lots of people there. The common theme is complex system modeling, but that cohort also includes, when they have time to attend, Axel and Franz. And this may be another point at which you can potentially engage in the nitty-gritty of how you actually model experimental data from it's usually fly mitochondria from different sort of cell lines with different sort of knockouts on on the cell lines. So I'm just putting that out there.
That's amazing. Just to interject very briefly, Carl, this is absolutely mind-blowing. It's fantastic. I did, with a subgroup of the people I'm collaborating with, make an application to ARIA, who currently have an opportunity space out called bioenergetic engineering, towards the kind of scientific, evidencing, endeavor you're describing around trying to understand if they're successful in editing the mitochondrial DNA, what effects would that have, and building the systems to try and explore and understand those effects to fit the data to the model. So I'd absolutely be delighted to join that DCM working group if you can help me figure out how to get an invite there.
Right. Well, I was going to say you just have to visit the website and email.
Super easy enough.
The problem is a friend of Tim Verblin, Magnus, tried that last week and didn't get a response, so we're trying to sort out the email. But at some point, Johan Medrano is who's a very brilliant but ex student of ours. Is basically the master of ceremonies and the convener of that meeting. And if you can't find it on the I'll email you the website and his email, and you can approach him. Just one little twist here. There's also another lady who's again an erstwhile student of ours called Noor Sajid. She is now looking earnestly at models of mitochondrial function from a translational perspective in psychiatry. So there is interestingly a sort of a multilateral theoretical investment in mitochondrial function, but this would be more the translational angle. It would not be the developing the principles for natural intelligence or artificial intelligence research. So that would be, if you like, a secondary route to engagement, in the fond hope that Axel and Franz are in attendance at this work group. It's sometimes very sparsely attended when they have to pick up children from school or there are holidays. Sometimes there are lots of people there, but that would be one avenue to make friends. And that you've heard of these people, so that's good. I just put this up here as a proof of principle. You can use the free energy principle to model intracellular dynamics of a realistic sort. And the game here is just to spot the functional forms or the isomorphisms between those functional forms that allow you to interpret electrochemical dynamics in the cell in terms of a gradient flow on variational free energy. In this particular example, it was all about dendritic self-organization and sort of dendrites sampling in the right way their environment, where their environment were all the sort of potential presynaptic inputs that make sense. So it's not a million miles away from the mitochondria talking to the matrix, or you know, to the intracellular cytoplasm, but it doesn't. Its focus was not on the. I repeat, the sort of the oscillatory, or you know, the phase relationships of you get in generalized synchrony. But again, if you just wanted a proof of principle that there are ways of casting microscopic intracellular self-organization as free energy minimizing processes, that then equips your intracellular dynamics with a teleology that can be read in terms of inference and learning, that then justifies a translation of that into artificial intelligence research. That's one of a very early example. I think it's supposed to be like
Super.
more
Absolutely great template. Yeah.
Yeah, I just put this up just to reinforce the, you know, what you were saying about clocks. And
Yes.
well, you know, I'm doing this in reverse order, but certainly you know, any sort of computer science take on computation read as some kind of belief propagation or variational message passing in the spirit of the actor model of in computational science is gonna have to speak to reactive message passing. And certainly if you're going not neuromorphic, but if you're thinking about processing in memory, for example, that reactive message passing has to be scheduled in times, or at least in ordinarily in time. And you know, there is quite a large literature on this now. I haven't got a website or a paper, but you might want to look at the work of RX Infer.
RX Infer. Yes, I've been following them, and
all right, good.
I think I met Bert also out there in Holland.
Oh, right, of course, yes, that's his neck of the woods, isn't it? Yeah, so I think he's probably the best person in terms of developing active inference free energy minimizing schemes with reactive message passing. His work, I think, is in Julia. One thing which they have yet to embrace is not just the clock at any one level, but the separation of temporal scales in deep generative models. So if you want to tell a story that have any true AGI, is just a statement you've got to have the right generative model for the content or the ecosystem in which you are computing or you're interacting with your users. And that right generative model has to have this scale invariant aspect that also includes time, which basically means that the fast stuff is small scale, running very, very fast at lower levels. And as you ascend the depth of these generative models, things also slow down. And that means that the message passing between level 1 and level 2 in any deep architecture is really interesting, because it means you've got to do a lot more computations, so more, if you like, periods of an oscillation.
Lower down in the hierarchy.
Lower down the hierarchy for every wave in time, for every period duty cycle at the higher level. And this is a phenomena which I think is really important for any kind of intelligent processing. You know, that breaks the Markovian aspect. You'll find this in hierarchical Dirichlet models, and in a sense, the whole point of the whole reason for the success of transformer architectures is they reach back in time as well, but they don't have the deep structure. That will be the next step. It's when you've got now when you've got a generative model where there are slow contextual oscillations that now supply empirical priors or inductive biases on the control parameters of the fast dynamics at the lower level, and so on all the way down. And for you, of course, all the way down is right to the mitochondria.
Yeah.
So I think this is an important thing to sell, is that the sort of the dynamics in terms of oscillations and cycles is crucial, but also has to be seen in the context of a renormalization group that you mentioned, that is implicit in any fit for purpose generative model, which speaks to now unanswered questions in the computer science in terms of reactive message passing, that you have to wait until your lower level has gone through all its computations before you receive a request, and then respond slowly with an inductive bias or an empirical prior or some context that then coordinates and from in your language supplies the right control parameter contextually for the system at the level below.
Are you sorry to interrupt, Carl, but are you suggesting that the limitations of the current approaches is that they are sequential in nature? Because I remember, from Gordon Pask's work as well, this concept of concurrence is kind of an axiom of his conversation theory. So it is parallel interaction between the layers.
Yes, I don't know. Forgive me. I know lots of things about a small field, but I have no idea what conversation theory means. If it's parallel generalized synchronization at the same time scale, then I'm not talking about that. I'm talking about a separation of time scales where slow stuff provides a context, very much in the spirit of an adiabatic approximation in quantum, for example. And just being able to deploy that adiabatic approximation, which is basically the basis of this kind of reactive message passing in these deep temporal models, requires you now to think very, very carefully about the intrinsic clocks at each level and how they are aligned across different timescales, not within a timescale.
Understood.
So I'm not sure. It may well be that sort of correspondence is part of conversation theory. If there are, if the conversation is cast in terms of semantics as opposed to lexical structure, and then lexical structure as a phonological structure as opposed to lexical structure, and right down to sort of time frequency fluctuations in the in the acoustics. So there may be that part aspect, but it was more the vertical. If you've got separation of timescales in a universe, in the world, that now is installed in your generative model, what implications does that have for the message passing between one level and the next level? And it's not thoroughly addressed in computer science, and it's not thoroughly addressed in CRUD or actor models. Because they just send a response when they get a request, and they don't know when to send. What are the criteria to send a request? And what I'm saying is the criteria for lower levels is I've got to do lots of computation before I speak to my boss. My boss just needs to know the summary of the past month. He doesn't need to know the day-to-day developments. And the boss likewise will not micromanage. They'll just provide a contextual guidance. Oh, this month you'll do that. But the coordination now means that there is no clock time, there's no universal time. It's just a question of how many operations do you do before communicating with the level above.
But then you also have the frame problem to deal with there. Do you not?
Yes.
Yes. Yeah, which is why there has to be some kind of dynamical interaction between these synchronizing clocks across the hierarchy.
Yes. Yes. No. Yeah. We'll use the notion of a frame problem, but specifically in this instance in time.
Yes. Yes. Exactly.
Oh, sorry, that was it. It was in neuroscience and biobehavioral. Sorry, it being just something to remind me to talk about the importance of separation of temporal scales. But you know all this because you read the renormalizing generative model paper, which
Yes,
sort
I
of
have.
takes a sort of renormalization group approach. But if you just wanted a more intuitive but neurobiological take on these issues in terms of communication, literally. then this is something with Thomas that might be
Excellent.
interesting. So yeah, another I'm just where to go. What I wanted to say was that certainly I was both impressed but also compelled by the repeated reference to modes of oscillation and phase reduction. And when I say phase reduction, what I mean is a mean field reduction, a coordinate transform in your parlance, to phase, an oscillation just in terms of phase variables, and the beauty of being able to work in with phase variables, or that particular kind of transformation, or what could also be cast as a sort of mean field approximation. I just wanted to make the point that it can't be any other way from the point of
No.
the energy principle, simply because, sorry, it can't be any other way from the point of view of the free energy principle, in the sense that for any system to be open and to possess a non-equilibrium steady state, you have to break detailed balance because it's open, and that entails solenoidal or non-dissipative dynamics. And as soon as solenoidal or non-dissipative dynamics, you've got the notion of some kind of phase or some kind of orbit. And I think that's absolutely central to the statistical physics of self-organization. So the free energy principle, even before you start sort of spinning off active inference, tells you something quite fundamental. However you're going to coarse grain your system, or describe your system, or model your system, or do a mean field reduction on your system, you're going to have to deal with orbits and implicitly phase in those orbits. So that may be, again, worthwhile foregrounding and explaining why there is so much focus on oscillations and modes and vibrations and resonances. Of course, if you take that to the ultimate limit and just want to do a appeal to dynamical systems theorist, you're just also you can also frame that in terms of synchronization of chaos, or generalized synchrony, and that is the variational free energy minimizing solution. So the minima of the variational free energy live on the synchronization manifold that engenders synchronization of chaos. And of course, synchronization immediately on the tin says, well, you've got, what is synchronized? Well, it has to have some oscillatory or phase phase attribute. So this would be more the parallel sort of within a level coupling between the dynamics on the inside and the outside. So there are many instances of that, and what that sort of the perspective afforded by synchronization of chaos, or generalized synchrony, tells you something really interesting. It tells you that both the inside, the computer, and the outside, the computed, or the observer and the observed, both have to have non-dissipative dynamics. And I always smile when I think about this, because I'm thinking back to Prigogine and dissipative structures, but it's not the dissipative part that's interesting, it's the non-dissipative part that is interesting. And where does that come from? Well, it just comes from autonomous dynamics that have an attracting set. And you mentioned the Lotka-Volterra system, generalized Lotka-Volterra one, and that made me smile, simply because every paper we have ever written illustrating an application of active inference or the free energy principle to biology has always started with either a Lorentz attractor or a Lotka-Volterra system.
Right.
You can't do it any other way.
No.
And this is my favorite. It's a very early example, but it's my favorite example. Here we are, using a Lotka-Volterra system to generate predictions that were, in fact, in this instance, enacted through sort of moving the hand and writing. But the point being made, I just wanted to emphasize here, is the central foundational importance of having orbits as part of your generative model, because you have to synchronize with the world. Why? Because you have to have solenoidal or non-dissipative dynamics. Why? Because you are open to the world, and that would be the definition of breaking detailed balance, or just having a non-equilibrium. Just having a non-equilibrium steady state means that you have to have that sort of solenoidal part in play. The technical discussion of that, if you haven't already read this, is unpacked in this treatment of stochastic chaos under the free energy principle, just using the minimal system, a Lorentz system, in fact, two coupled Lorentz systems, just to dive down into the real importance of this non-dissipative, conservative, solenoidal dynamics that has the attribute of phase and has the notion of an orbit, and how even with the simplest kind of chaotic dynamics, or you can now interpret the dynamics in terms of inference and possibly learning in the fullness of time. So I thought if you haven't read this, this would be an interesting mathematical treatment that sort of just foregrounds the importance of decomposing dynamics into dissipative and non-dissipative parts. This gentleman has also done some interesting work on, but no, that's a diversion too far. So yeah, we've done.
I'm
that.
soaking it up. Don't spare me.
All right. Okay. Yeah. So this notion of free energy minimizing solutions being definitional of existence, but also being having this sort of inference and learning or intelligence interpretation. The casting things as generalized synchrony has emerged in many parts of demonstrations of the free energy principle. I just put this up because it speaks to the emergence of generalized synchrony in networks of mutually inferring neurons, which would be, I would imagine, have a very, very simple functional form to connections either of on a ring, on the ring that you described, or indeed having multiple mitochondria, or indeed multiple cells together, or cellular populations. I think the principles with just local or lateral interactions engendering the generalized synchrony is it couldn't be any other way, and this particular example just pursues it in terms of Markov blankets, if I remember. But there are lots of other examples that you could refer to. The point I repeat here is that if to exist you have to minimize your free energy or maximize your marginal likelihood, then it must be the case that you will evince some generalized synchrony. There will be a synchronization manifold between the thing and everything else, and that sort of means you know where to look in terms of the functional forms of how you might want to coarse grain or model these systems. The other interesting thing that the other interesting aspect of that that kind of generalized synchrony was in terms of your discussion about keeping close to bifurcation and inducing the sort of critical slowing that you get in self-organized criticality. There's an interesting take on that from a mathematical perspective, that if you take any random dynamical system that is separated from its heat bath on the world, and just cast the dynamics as a gradient flow on free energy on this synchronization manifold, then you think about the functional form of the free energy, and you note that it has an entropy maximizing aspect, which in accord with James's maximum entropy principle. And what that means is that the as you're moving around in this geometry and this information geometry on this statistical manifold, that means that the curvature is at the free energy minima always very small, because the curvature corresponds to the entropy. So if you've got very low entropy, if your current dynamics, your sort of mitochondrial state is encoding some probability distribution over some external states, so the intracellular, the non-mitochondrial part, then it's going to have a very high curvature. As you get to the minima, then you find you get basically flat minima that have a very low curvature and therefore a high posterior entropy or observer entropy in the spirit of jet. That's interesting because then the dynamics on this low curvature small Hessian manifold have eigenvalues that all approach zero from below. What does that mean? It means you get critical slowing for free and the emergence of modes that don't decay very quickly. So by construction, by definition, generalized synchrony at free energy minima will always show a kind of self-organized criticality of a very simple sort, where the eigenvalues associated with the dynamics, the eigenmodes of the dynamics, are generally approaching zero from below, because the Hessian or the curvature basically determines the nature or the size of these eigenvalues. To my mind, there's a lovely link now between self-organization, dynamics, eigenmodes, and self-organized criticality, which it couldn't be other way. So often I'm asked to review papers on self-organized criticality, and people say, "Isn't it a remarkable phenomena?" And I always think to myself,
can't be any other way.
Exactly. Show me something that doesn't show self-organizing criticality. That'd be interesting. The second reason I show you this one is that you talked a lot about the control parameters that manage that self-organized criticality. And of course, because the curvature of the free energy landscape depends upon the posterior precision, it also depends upon all the parameters of your generative model, but there's some very special parameters that have an enormous effect. And these are the sort of inverse covariance of the precision parameters. And I say that because the precisions, and this is shown in at the end of this paper, in terms of what would happen if you start to change these precision parameters, and all sorts of interesting things happen. So perceptual categorization, when you find that Goldilocks regime in the edge of chaos, with a very the principal exponent now attaining approaching zero from below, a very low free energy with lots of critical slowing. In this regime, you've got a nice balance between the precision of different parts of your generative model. And if you break that by having the precision go too high or low in relation to another part of your generative model, then you lose this. So it is the case that you've got this, it's really important to get some control parameters right to get to keep you in this free energy minimizing zone. And if you get it wrong, then you are no longer in the minimum free energy, but also you lose that self-organized criticality. I say that because the precision at the level of the dynamics is a rate constant, and it speaks again to the importance of dynamics. But in another setting, the precision also has some really interesting interpretations in terms of both machine learning and also translational research. So precision is usually read as, or optimizing the precision, getting the precision right here is usually read as selective attention in psychology. And of course, that is easily linked now to attention heads and transformer architectures. So the whole reason that transformer architectures work is because they've got this sort of effectively a precision control, a Kalman gain if you're a control theorist, or attention head if you work with transformer architectures, speaking to a really important kind of control variable, although that's not actually variable in transformer architectures, but in generic architectures, you know, it strikes me that's a very important control variable. So there are two reasons just to put that paper up, just to motivate the sort of story about self-organized criticality, but also foregrounding particular control parameters that have an enormous impact on the on that self-organization. And this was demonstrated in in one of many ways when it comes to communication. So again, in terms of selling this in terms of federated inference or learning in machine learning. This paper was just about establishing generalized synchrony between two things that were talking to each other. In this instance, two birds that were singing to each other, and the importance of the precision of one bird in relation to the other bird in terms of master-slave or skew product systems. And what tends to happen if you get this precision control parameter, if you have very precise beliefs, then you're going to pull your student, your child, your slave towards you during the emergence of the generalized synchrony, which means effectively you're teaching the student, the slave, and the child, simply because you've got more precise beliefs, because you're more confident in terms of deploying your generative model. So, you know, again, it's a long way from mitochondria, but it does speak to multiple ways in which the underlying principles of generalized synchrony and phase relationships unpack at every level, and how they depend upon key control parameters. I'm trying to find an example where, no, my apologies. I was hoping to show an well, perhaps you can see it a little bit. This is the imprecise one, and this is the precise one. So what you're meant to be seeing here is the imprecise, the baby bird approaches the adult bird. So it now learns a generative model, so that they're now in synchrony, and they've both got this sort of have access now to this joint self-organized criticality that keeps them on that free energy minima. So that may or may not be useful, but it certainly has lots of nice oscillations.
It's fascinating, absolutely fascinating. I'm just thinking, is that that's a transitory free energy minimum, presumably, because as you say, if you're a master slave entrainment, you're learning, the generative model of the master, which is not necessarily the optimal landscape or the optimal saddle to reside on. So
yes, that's well spotted. Yeah, you're always chasing a minima. And of course, there is no minima. You're always going downhill, but it's a moving target. Absolutely. And interestingly, if you just think about what that means for the sort of oscillations in the orbits, I mean, strictly speaking, the solenoidal part, or the conservative part, is actually moving on the isocontours of the free energy landscape.
Yes.
But of course, in so moving, the movement is now going to be informed by the data. So the free energy is a free energy function of the data, and so the data are constantly shifting your landscape. So your landscape itself may or may not have a nice little global or local minima, but as time goes on, it's moving. So you're always chasing it. In some way.
This is the dynamical nature of expected free energy is minimization as well, presumably.
Yes, yeah. So you have to roll out into the future in order to compensate. It also emerges in a control theoretic setting in terms of encoding not just the current state, but the little path into the future by moving into generalized coordinates of motion. So a lot of the complex system modeling that we do works in generalized coordinates of motion beyond second order. So it's beyond, so I'm not talking about generalized coordinates in terms of P and Q in physics. I'm talking about generalized coordinates of motion. So we're talking about x dot, x double dot, x triple dot, usually up to about 4 orders of motion. And then once you've got a sort of this generalized state space representation, you've effectively got a little representation of a little path into the future, because you use a Taylor expansion of those higher order derivatives to work out where you're going to be in the future and where you've been in the past. You accumulate uncertainty very quickly, so it's just a local path, but it still stands in for a pathological formulation. So, but that's just a useful technical device. I don't think you need to worry about that at the moment. Although one could, if one thought about putting your model into a dynamic causal model of the empirical data that Nick and Mike's students are acquiring, but perhaps that's a conversation for another day.
Carl, thank you. It's been tremendous. I really appreciate your attention and this incredible rich landscape you've described in terms of those papers, and for your support. Thank you very much.
Oh, thank you.
Oh, thank you very much, Carl, as always. And thank you so much, Anthony. To give a round of applause for Anthony. Very nice presentation. Thank you so much. And
thanks very much, Miguel. Thanks, everyone.
See you, everyone, next time.
Bye-bye.
Bye-bye.